to talk on decode computational and metabolic insights
molecular
into
mapping. If I would like to, since there are many
anti-no-bacterial-mediated
students are here, I would
measurement
like to
for
spend a few
PLB.
minutes in this
Thank you.
particular slide. Whenever you undertake molecular mapping, the first and foremost task
You have 10 minutes,
is in terms of
Roy.
identifying a good source of resistance. Preferably it should be monogenic, and you need to identify a variety which is susceptible to that particular pest or disease. Then you develop a mapping population. The most commonly used mapping populations are F2, F3.
Good afternoon to everybody
advanced
present
up to
for this
F5 or
session.
F6 generation.
Today
by
I'm
which
going to
time
talk about the
they will,
computational and
become,
metabolic
both,
insights into the actinobacteria
they'll be heterozygous
mediated
genus,
management
but all
of
lines will be
bacterial leaf
homozygous.
blight of rice.
Individual plants in the
Rice
line will be
feeds
homozygous. But
half
there
the
are
million
alternative rules
population of the
wherein we can
world.
develop
And a
backcross
major
inbred
hindrance
lines
to the rice
or
production in
near-residenic
this
lines.
world is
Once you have
caused
a
by
mapping
bacterial leaf
population,
blight of rice,
a
which is
stabilized
caused by
mapping population is always
Xanthomonas
good.
oryzae,
When I say stabilized
and
mapping
you can
population,
see that
I'm talking
chloropet
about
map in
generations
West
like
India comes
beyond
under a
F5,
high
like
risk
F6 or
zone
F7.
for
And when it is
bacterial leaf
backcross
blight
derived line,
susceptibility.
it should be at least BC1 or BC2,
As the world
F4
is
or
shifting
F5
towards
generation, wherein the trait will
sustainable
not segregate any
management,
further.
there is a high
So
need
once we
in the
have
hour
a
for
stable
the
generation, we have to go for
biological control
large-scale
management of rice.
phenotyping, high
And
throughput
why
phenotyping,
actinobacteria?
wherein
Because they are
each
a
and
very
every
good plant
should
growth
be
promoting agents,
every line should
and
be
also they
phenotyped
are
at least for
elicitors
2
of plant
continuous
defense,
seasons.
and also they are a
And
very
once
good
phenotyping
biological
is
control
done,
agent.
we have got lot of options
Isolation
in
and
terms
enumeration
of
of the
genotyping.
actinobacteria
And,
from rice
I would
rhizosphere
not like to
and
go
the
into
screening
what
of
all
those
the different
actinobacteria
markers or
against
steps
the
used there.
Xanthomonas
But
oryzae,
at present, the most preferred marker
potent
system in rice
actinobacteria,
for
which are
genotyping are
screened
SNP
through the
markers.
primary and secondary
Different
screening
platforms are
were further
available in
tested
rice. If
in
anybody is
glass,
interested,
house
you
and
can
field
write to me. I'll
condition,
explain you in detail about
metabolomics
which are all the
profiling of the
phenotyping
potent
genotyping
actinobacteria
platforms
using the
available for
HRCMS
genotyping in rice,
method
particularly
and the
SNP
molecular
markers.
interaction of the
And then once
pathogenicity
you
factors of
have
soup
a marker
with the
system used for
actinobacterial
analysis of
metabolites
this,
in in
say,
silico
for a
conditions
fixed mapping
under
population,
molecular dynamic
then you develop
modeling.
a linkage
These are
map
this is
using
the
those
flow
markers.
of the
Even though
study.
those markers, physical
These are the
positions will be
diverse
known already, but
actinobacterial
still,
isolates
this is
from
the
the
first
rice
and foremost
rhizospheres,
task which you have to
isolated
do it.
using the serial
Then
dilution
finally, you have
method
to
in
go for
starch,
QTL or
casein,
gene
agar
identification
medium.
and undertaking
And primary
course
screening
mapping,
of the
and then with a
actinobacterial
subset of that
isolates was
particular
done
mapping
using the
population, you can
dual
do
culture
intercrossing
plate,
and then go
and
for
the
fine
8
mapping.
isolates were
And,
found to be effectively
even though it's not essential to
reducing the
identify
disease suppression, and they were
the
taken for
exact location
secondary
of the
screening
gene
in disk
and
diffusion
identifying what
assay,
is the
and
causative gene
AMGD
underlying that
16,
particular
20
trait.
and AUDT
It will be
502
desirable.
were
That's why
used to
most of
were
the
very
cases what I'll be
effective
going to
to
describe
control
as of
the bacterial leaf
talking
blight
about
of
molecular
rice.
mapping. We didn't
And
just stop
molecular
at
characterization
course
of the
mapping or fine
AMGD
mapping.
16
We
was
went
found
up to
to
the
be
gene level, not only
Streptomyces
in terms
ginkgonus,
of
and AMGD
fine
20
mapping
was
them, but also
Streptomyces
characterizing
zyamensis,
them
and
at
when
functional
were
level through
biochemically
genomic,
characterized, they
transgenic, and also
were
transcriptomic
producing
approaches.
siderophore.
And finally, when you have a
positive for
closely
IAA
linked
production,
marker, in the
catalase
case of
production, and also
even
HCl
if you don't have
production
a
and
closely linked
phosphorus
marker, you can always have
solubilization.
two
And when
markers
taken
flanking
under
the gene,
field
which can be
evaluation,
used
the
together.
seed treatment
And
at
if you
10
have
liter
a
per
marker
kg
which is
and
part
2
of the
foliar
gene,
spray
these
of 10
markers are
ml per
functional markers.
liter
An
with
example was
AMGD
presented by
16
Dr. T.R.
at
Sharmaji
10 raised to
for
8 CFU per ml
gene.
had
And once
enhanced
you
the
have a
root length,
good
the
marker which
plant
is very
height,
closely linked to the
reduced
gene,
the
or two
lesion
markers
length,
which are flanking the
and
gene,
which
or
showed
marker
that they
which is
are good
part of the
plant
gene, which are
promoting
called
and
as
disease
functional
suppression
markers. We
agents.
can go for large-scale
The
marker state
AMGD
selection
16,
using various
which was a
tools or
very
techniques.
good
And
biological control agent,
how
was
we are
further
going to develop
taken for
different
HRLCMS
methodology
analysis
to transfer the genes, I'll
understand
be
its
discussing
metabolome.
later.
And
Now coming to
based on the
some of the
area of
own
abundance,
success stories
different
or with regard to
metabolites were
bacterial blight
selected,
resistance,
and
my team together
literature
with
mining was
Dr.
done to
Laha,
understand the
Gaurishekar
function
Laha,
of those
and
metabolomics,
with the
and
help
we
of my
saw that there
pathology
are
team, they have
different
done very
indole
good work in terms of
derivatives like indole 3
identifying.
acetic
This is
acid,
one of the
indole
genes
3
which
carbinol, methyl
which has
indole
been
3
used as a flagship gene
acetate,
in
and also
many of the distance
indole 3
breeding program.
acrylic
It's a gene
acid
called
and
XA33.
pyroxidin. They are all
It was course mapped using
very
SSR
good growth
markers on
promoting
chromosome 7 of rice.
metabolites, and
But
also
recently,
lumichrome,
one of
fusidic
my
acid,
PhD
and
students, he has done
vancomycin,
the fine mapping,
oleomycin. They are
and
all
we have
different
identified the
antimicrobial
underlying candidate
agents
gene
which
also.
can be used for
But
further
since we were dependent
reducing
on
the
low
Xanthomonas
throughput markers like
disease suppression of bacterial leaf blight.
ISSRs, and then
This is the
RFLPs in
total
initial
ion
stages,
chromatogram
the
of the
progress was very, very
actinobacterial
long. It took almost
isolate
6 years
plotted
for
by
us
relative
to
abundance
undertake
against the
molecular
time
mapping,
retention
gene
time.
identification, and then
Different
gene
peaks show the
characterization.
relative abundance
But
of different
the scenario
metabolites,
with
and
regard to
several
this
peaks
has
show the
changed with
diverse
advent
nature of
genomic
metabolites
tools,
produced
which
by
are,
the AMGD60.
very well described by Dr. T.R.
And
Sharmaji. So now
in
that
order
we
to
have got
understand
very
where
good
these each
SNP markers
metabolites
and also
belong to
high
which
throughput
pathway,
genomic
the
tools,
global pathway
for
enrichment
candidate
analysis
gene
is done,
characterization.
and each
Once my
bubble
student,
represented
PhD
the
student
different
Pragya, she identified
pathways.
that one
And
of
the major pathways were found
lines
as valine
provided
using
by
isoleucine biosynthesis and degradation pathway, the pyrimidine metabolism, purine metabolism,
and
tryptophan metabolism. They
did,
all have
genotyping with
different roles
7K
in plant growth
rice
promotion
SNP
and also
markers available.
microbial
And she was
suppression.
able to identify a
And
major
further, to understand the molecular
locus
level
on
of
chromosome 11
interaction,
long arm, which is about 3
pathogenicity
kb away
factors,
from
that is
XA21.
PTHO1 and
she has also
peptide
worked,
deformulase,
to
which are
identify
very
the
important for the
candidate gene
virulence
also.
and
We
the
were lucky
viability
because
of the zone
the
were
underlying
taken,
SNP was part of the
and
gene,
the molecular
we
docking
were then
studies was
able to,
conducted between
through
the
sequencing and
different
also through
metabolites and also the
transcriptomics and then later through
pathogenicity
transgenesis and gene
factor. And
silencing, we were able to
PTHO1
identify
binds with the
that
promoter
the
region of
candidate gene
OH
is a
sweet
gene
gene
encoding
11,
NBRC
which will
domain containing protein.
upregulate and
This
lead
is
to
another
the
success
sweet
story wherein my colleague Dr.
sugar influx.
Sesha Madhav,
And this is the
together with
Ramachandran
Dr.
plot
Srinivas
showing the
Prasad's team,
backbone
they have worked towards
dihedral
identification
angle
of,
distribution between the phi
major,
and psi
leg
angles
blast
of the
resistant
amino
gene.
acid
It also
bases,
confers,
and most of the
resistance in leaf
amino
blast
acids
also.
were in the
This
allowed
was
and
identified
disallowed
from
in the
wild
favored and the
species
allowed
called as
region.
Oryza
And you can
glumipetala.
see that
The
lumicrome
introgression
has
lines were
bound with
developed
a
by PAU,
minus
Ludhiana.
6.6
So he
kilocalorie,
worked
binding
initially
energy of
with
minus
SSR
6.6
markers,
kilocalorie
and
per
later
mole,
with the high
and this
throughput
had the
tools like
least
SNP
binding
markers,
energy, and this
genotyping
was
by
and had
sequencing and other
three number of
advanced
hydrogen
tools which are
bonds, which was
available.
further
He was
taken
able to
for
identify the
dynamic
candidate gene,
modeling
and
to
this gene
understand the
has
stability
been
of the
licensed to at
protein.
least 3
The
or 4 companies
RMSD
for
plot
deployment in their
also
breeding
shows
programs.
that in the initial phases there was
And this is the power
fluctuation,
of
but
mapping.
towards the
So
100
right from
nanoseconds, the
the day in which
entire
mapping
complex
population
was
was developed,
stable.
within
And when
about
we had
1.5
seen the
years, we
RMSF,
can
that is
easily
the
undertake
root
or
mean
identify
square
the candidate gene
fluctuation,
and validate
only
it also.
controlled fluctuation
And,
was there, and the
this
radius
is
of
one
gyration
recent
also
example. I
showed
mentioned that
the
sheet
entire
blight
protein
and false
structure was
spot are gray
compact,
areas.
and the more
And,
number of hydrogen was
they are not having
bond
very
was
strong
found
level of
towards
resistance.
the
Of course, there
entire
are a few genotypes like
like entire over time, which shows the
level
stable
of
nature of
resistance
the
is
complex. And the
towards the
superimposed
moderate
structure also
resistance side.
shows that
So
it
we
is
have
an induced fit
identified a
model, which
northeastern
shows the
land
stability
race
of the
by my
protein
colleague
and the
Dr. Vijaypal Badana, who
ligand.
earlier
And further,
worked at
PTHO,
Nagaland,
the
Manipur
peptide
and all. So he
deformulase
brought this particular line. We
protein also
developed a
was
mapping
taken,
population
and
by
here
crossing
also
it with
the
improved
stability, the
Sambhav Sudi.
stereochemically
So we had
stable
a
protein
set
was
of
present,
430
and here
RALs.
also
So
lumicrome
using
had
that
bound with the
step markers, with the help of
peptide deformulase with a
NAPGR,
binding
Dr.
energy of
Gopalji
minus
Jazz group, we
8.3
were able to
kilocalorie per mole.
do a
And
fine
here
mapping
further you
using
can
step
see that
markers. And
the
we were
RMSD
also able to
plot,
identify diagnostic
RMSF
SNPs,
plot,
and we are now
all
identified
the
3
plots
putative
show the
candidate genes.
stabilization of the protein.
That validation.
And the
this
superimposition
work,
showed a
I would say that will get
induced fit
completed
model.
within a time period
The
of
key
1 year.
outcomes are
This is the power of genomics. This is the power
AIM-16,
of
acid
deploying
treatment and 2
advanced
foliar
tools in
spray
molecular
reduce the
mapping.
bacterial leaf blight
Then
incidence to
this another typical case
32.17
wherein my colleague Dr. Jyoti
percentage,
Badri,
and the
she works on
lumicrome
population
effectively bound
improvement
with
with
the
regard to sheet
pathogenicity
blight.
factors
So from a
of the
northeastern land race
xanthomonas,
called
and
Fogger, which
lumicrome
shows
can be
strong
considered
level of
for
tolerance
the
to sheet
future
blight.
applications
She has
can be
identified
lumicrome can be
a major locus
suitably
called
on commercial scale.
SBR 3.1 on
Production
chromosome
can be
3,
undergone
and,
through
she's
microbial
working towards
fermentation, and it
identification
has
of the candidate
both
gene.
antimicrobial and
we
as
have
well
been,
as a growth
successful to some
promoting
extent because two
character.
genes show hyperexpression
And
after
till now, no
inoculation with the
direct
pathogen.
Antimicrobial
And,
agent. Like
similar is the
lumicrome was
story
never
with
used
regard
as an
to,
antimicrobial
another,
agent in
gene
plant
conferring
disease
tolerance to
management.
BPH
So this
called
is
BPH
one of the
33.
studies
The
where
original
the
donor
importance
is
of
from
lumicrome is
land race called Veltachira. From that
well
breeding line
documented,
called
and the
RP2068
structural
was earlier developed.
stability is
We
being shown
identified
through
that this
dynamic
particular
modeling
line shows strong
and,
level of resistance against
molecular docking.
BPH.
Thank
So we developed a mapping population. This program was led by
Thank you, Ms.
Dr.
Shita Roy,
Ventur.
for an excellent
So,
overview and
with
also
his team, my
sharing some
team has,
insights on
initially done the
actinobacteria-mediated management
post-mapping,
of
then fine
rice
mapping
BLB.
with
Thank you so
markers.
much.
And we have identified that the
quick
candidate
questions
gene
to Ms.
controlling
Ashita
this particular responsible for this particular trait is a heat shock protein.
Thank
and
you.
now we have developed candidate gene
I think
specific
last
markers, and,
3 hours
we are in
we
final
talked
stage of
about
varietal
and
development
we
with this particular
heard a lot
new
of
gene.
story or
And,
findings on the rice. Now
one
we
important
have
power
2
of,
next presentations which are
like say,
on
we have been talking about
one is on brinjal and another one on
cross-steroid mapping
sorghum. So first I would like to
Other
invite
popular methodology is
Mr.
called
Nikhil
as
Kumar
association
to
mapping,
talk about
wherein historical
decoding the
populations
metabolites during the food development for
crossbred
disease
developed
tolerance
populations
in
larger in
brinjal.
size, around 100 or thousands. Generally, they are used for trait
Good
mapping,
afternoon, everyone, and
phenotyping,
all
and then
present here.
validation.
I am happy to
Through
present
association mapping,
one of the
my
interested
colleagues
crop
Dr.
other than the
Kalyani and
major
Dr.
crop.
Somashekar,
Here I will
they
be
have
presenting
indeed identified
the work,
some
our
major
work,
loci
decoding encoding the
located on
metabolites
chromosome
during
1,
the fruit
2,
development
4,
for
and
disease
8,
resistance in
associated
brinjal,
with,
Solanum melongena.
rice root
Basically,
knot
eggplant
nematode,
is
which
widely
becoming a
cultivated and
serious
economically
problem,
significant
particularly
crop in
upland rice
Asia,
and
and
also
like
direct
in
seeded
India also
rice.
it is like
This
major
was
crop
done
used as a
using our
food.
eCrypt network,
And
wherein we have
it is
started a
susceptible to
coordinated
fungal,
testing
bacterial,
program even
insect
for
pests,
nematodes also.
and it
With
leads
the support
to
of our
yield
cooperators,
loss and
we are
reduced
able to
marketability.
achieve this. And right
The
now we
secondary
are
metabolites
working
like
towards that
phenolics,
major
flavonoids,
locus located
anthocyanins,
on
glycoalkaloids
chromosome 4,
not
short
only
arm,
contribute to
and
taste,
we are working
but
towards
also it
identification
plays
of the
an
candidate
active
gene
role in plant
with the
defense against
biparental
biotic stress.
cross-derived mapping population with
Defense
two donors.
responses like
Then this another
metabolite accumulation depend on
I
developmental
would say
stage and tissue
a
type.
success of coordinated
This
research.
untargeted metabolite
In coordinated
captures the
research
global changes
under
in
the guidance
defensive
of
metabolites,
Dr. T.R. Sharma
which helps to
starting
identify
with
the
the year
biochemical markers
2023,
and
we have
pathways
been
involved in food
fingerprinting
defense and
each and
disease
every
resistance.
entry nominated
This
for
is the one special
our
variety
Eclipse speech,
called
IHRT or
Matu Solana
IVT or
melongena variety
AVT1,
Matugula, which is only grown
Every
in
year
Matu
we
village of the
fingerprint
Udupi
around
district,
1,200 to
which bagged the GI
1,500
tag in
entries
2011
using
from
1K
Chennai.
rice S&P platform. It has got representation of
For
around
this experiment, we
1,084
have
S&P
selected
markers.
3 fields
One
in which
lucky thing what
we have selected
got out
25
of it
plants.
is I
In each
requested
plant, we
Padma Kumari
labeled
madam
5
to
flowers,
analyze
and
the
later,
data with regard
in
to
the time interval
stem
of
borer
15 days, we
because
have collected
we have
the
a stem borer
samples.
screening nursery carried out every year, wherein
Later,
all
the
these
peel and pulp were
1,300 lines
separated
are
and
subjected
metabolite
for this
extraction
thing.
were
Then
performed
she
using acidified
carefully
methanol, followed by
selected some certain
homogenization,
individuals and then
sonication, and
developed a mapping
concentration.
population which doesn't represent the
This is
structure.
this table 1
And my
represents
colleague
the
Dr.
gradient
Kalyani,
program
who is
for
a genomics expert,
LCMS and the
could
instrument
do an association
parameters.
mapping
And this
using this 1K RICA.
that is
And
that
we were
figure
surprised
4
to find
represents the
very good
workflow
associations,
which has been performed in
particularly
the
on
compound discover software.
chromosome 5, wherein we
This
got
figure 5
one
represents the
single marker which has
multivariate
got a
analysis
large
for the
peak.
15 DPA peel and pulp samples, where we have found 400 plus metabolites, in which we have found 123 metabolic common metabolites, where
I would say that
44
future
metabolites
lies
were
also in
upregulating and
association
47
mapping.
metabolites were
And
the
we are going to
downregulating.
use our All India Coordinated Research Project
Whereas in
on
30
rice
DPA,
data
we have found that
significantly for
330
association
metabolites,
mapping.
where 77 metabolites
And,
were found in
I
both
would
peel
like
and
to
pulp,
stop
where
here
10
with regard
metabolites
to,
were
only
upregulating
given
and
a
19
glimpse of
metabolites were
molecular mapping because
downregulating.
of paucity of time. If any student
In
or anybody
45
is
DPA
interested, he can
samples,
always write to me,
whereas the
and I'll be happy
pulp
to
sample, we have
assist
found
you
that
in
more
various,
metabolites, and for only 45
stages in which you
metabolites, we
request
have found a common
help.
in both.
Now
And
let's talk
compared to
about
the initial
how-
stage of the
what are
development to the
markers or
final
what are
mature
major
stage of the
genes for
development,
which molecular
the
markers
common
are available,
metabolites are
how
reducing and the
they can
unique
be
metabolites were
translated
increasing.
by developing
So
better resistant
we thought
varieties or
to check
hybrids.
what are the major
So in this
disease
regard,
resistant metabolites and the
there are
metabolites
several breeding
related to
strategies.
defense.
One
So
breeding
we
strategy which has
performed
been
that
very, very
analysis, and we have found
popularly
that
deployed for
here we
resistant
have compared
gene
in between
breeding
the
is
stages.
backcross
Previously,
breeding,
we
wherein
performed the
generally
analysis
the
among
background
the
of a very
like,
alike
peel and pulp of
variety
the same
which
stage.
not only has
Here
got
we
a higher
performed
yield
like in
but
between
also
stages.
a good grain and
This
cooking
slide
quality.
represents the
We
disease
try to
resistant
transfer
metabolites
a
in
few
field
genes,
samples,
maybe
like
3,
among
4
like
or
15
5. They will
versus
be
30
encoding a
DPA,
single
30
trait,
versus
or they
45,
could
and
be
15
encoding
versus
multiple
45.
traits.
Like we
So
have found
backcross
that only
breeding
few
or
metabolites which
marker-assisted
are found
gene pyramiding
common
is
in
one of the
like both
most popular
stages. Like
strategy.
the other
But
other than that,
of late,
there are the many
additional
unique
tools like
metabolites.
genome-wide association study, wherein
This
you
is the
don't
disease
look at
resistant
single
metabolites
genes
in pulp
which are
sample.
either
Here,
major or
when
minor
among the
QTLs.
development, the number
So
of resistant
there are
metabolites were
certain
increasing when compared
traits
to the
like, for example, sheep blight,
early
we
developmental
identified that
stages.
even though there are some major QTLs are there, but
And later we
even in
performed the
Vazopec and Fogac, they are
metabolite pathway
controlled by
analysis,
several
where we have
minor
found that
loci
the
also.
compounds
So,
which we have identified
genome-wide
for
association study will help you
disease
to
resistance,
identify
they were
the minor
undergoing like pathways like
QTLs,
phenylpropanide and
then help
flavonoid
us to
pathway,
transfer those
oxylipine and fatty
QTLs into
acid
the background
signaling,
of our varieties.
amino acid defense
Then we
pathway,
have
steroid
genome
alkaloid
editing.
and
I'll talk only
cyanogenic
about one
glycoside pathway, and terpenoid
successful example
metabolism.
wherein genome editing has been deployed for resistant
To
seed
conclude,
breeding.
in this study, a wide and
also be
targeted
briefly be
metabolomic
touching
approach was
populations
performed
like magic,
to identify
and also
disease-resistant metabolites.
we also need to understand that
a total
conventional
of
breeding is still used for
1,041
resistant breeding
metabolites identified in 3
deployment,
stages,
but there are
among which
various
like
challenges,
half of like
and
more
that's
than
why molecular breeding
45%
is
of
the
the
right way
metabolites
for us to go
were
ahead forward.
identified as
As far as
disease-resistant
bacterial
metabolites.
leaf blight resistance is
This
concerned,
fruit development
based
involves the
on
progressive
our
metabolic
intensive,
reprogramming, where
analysis
field
of
tissues
our
accumulate
Eclipse
defense-related
data,
metabolites
wherein
in
we have got
stage-dependent
extensive
manner,
phenotyping
forming a
information
chemical
about
barrier against the
various
disease.
NALs containing
These
different
tissue-specific
genes.
metabolic
For bacterial
profiles
blight
suggest active
resistance, it is my
metabolism, likely
understanding that
reflecting
a 2 gene or 3
differential
gene
roles of peel
combination
and pulp
from
in
the
defense,
genes listed
transport,
here,
primary and
exate
secondary
13,
metabolic
exate
process
5,
during
exate
fruit
21,
development.
exate 33, exate 38 and exate 48
These are
should
the key
give
references
durable
used for this
resistance.
study.
With regard to
And I would
blast
like to
already,
thank my mentor
Dr.
Dr.
T.R.
A.
Sharma
Muthuswamy
has
for his
very nicely
constant guidance
illustrated,
and support. I would like to thank
gene
Manipal Academy
combination
of Higher
like
Education,
PA1
Manipal
plus
School of Life Sciences for their
PE2 plus
infrastructure and
PE54,
facilities.
PE9 plus
And I'd like to
PE54,
thank
PE68 plus
SERB and
PE54, all
ICMR
that. In
for
whichever
funding to our
combination,
lab.
PE54
And this is our team
there,
working on the
it gives
broadest
good
goods of
level
the
of
plant
resistance.
biology.
This I can say with confidence because
Thank you.
based on our fingerprinting data of
Thank
all our
you,
equipment,
Mr. Nikhil,
more than
for
90%
sharing
of
some
lines
of
which
your
have been
work on
identified to
metabolic
show good level of
pathways
blast
and also
resistance have got
profile. Thank you for
the resistant allele
the
of
excellent work
PE54.
and sharing with
And
all
with
of
regard
us.
to brown plant
Any
topper,
question?
even
Yeah,
though we
please,
have
sir.
got a few genes which have been identified to work very well in the country, but we don't have
Macrocarpan.
very good
Yes.
markers for
It
them.
is
We have
called
got only
tomato
SSRs
brinjal and
which
also
you can
edible.
deploy.
The good
Yeah,
part
yes.
is
So we
it
are
is
deploying
edible.
as a start
And,
BPH33 in our
if
breeding
not
program.
immune, it is very
And
highly
similarly for
resistant.
Galmit, we have got
yes.
genes like
I would
GM3,
request you
GM4 and
to
GM9,
parallelly compare
of which
the
we are deploying
metabolites
the two
with the
dominant genes,
macrocarpan with
GM4 and GM9 in
melangina.
pyramid conditions.
And see
we
where
now
exactly
have
we
some
can
success
Sure, sir,
also
sure,
with
sir,
regard to
okay.
breeding for Galmage resistance. And in all these things, our target
Okay.
is
No more question. Thank you again.
variety
Thank you, sir.
which is
Thank you.
So
resilient not
I'll
only to a pest
invite
or
now the
disease,
last
but also to
presenter
multiple stresses.
for the
That's
today
our end,
session,
target of this
Ms.
particular
Ranjita,
program.
to talk
Now,
on
let me
identification
narrate,
of
a
marker
few
trait
important
association
things about
for fall
how we develop an
armyworm
improved version of
in
Samba
resistance in
Masuri,
case
which
of
has got
sorghum.
durable bacterial blight resistance. And even as on today, the area under this variety is more than 9 lakh hectares. And there are farmers who have been growing this at least for the last almost 12 or 13 generations. And if you look into why this program started, this was a time when
Good
I joined
afternoon,
IARR.
everyone. I will
There are
be
a group of
presenting on the
farmers, who are
topic
from
identification of
Rayalaseema
marker
area,
trait
that is Nandyal
association
area, and
for
also
fall
from East
armyworm
Godavari.
resistance in sorghum.
They grow
This
Samba
study
Masuri
focuses on
regularly.
identification
And
of
they
the
expressed in
genomoid
1998 it happened when I joined
SNPs
IARR.
and their
They
association
expressed
with
to
fall
my head of
armyworm
department,
resistance.
Dr. N.P. Sarmaji,
This pest is
that we are facing severe problem of a disease called
recently
bacterial blight.
invaded
Then,
across the
in
globe.
collaboration with Dr. Ramesh Sonti's group, who have been a
So fall
pioneer in this
armyworm,
particular regard,
Spodoptera frugipeda,
we
is native
together
to
drafted
America.
a NATP
In
project of
2016,
ICR,
it was first
and then we
reported
worked
outside
to
its
deploy
native
backcross
in
breeding
Western
for
Africa.
development of
Then
an
in
improved version of Samba
2018
Masuri.
in India,
And in this, our target was that
at Karnataka.
we want to develop
So
a version
till now
of
it
Samba
has
Masuri, which
invaded more
looks
than
like Samba
80
Masuri,
countries
which
across
eats
the
like
globe.
Samba Masuri, and
With
which
this
cooks like Samba
rapid
Masuri.
dispersal
And
ability,
it should also have the resistance.
it
We should
has
not lose
become a
anything from the background with regard to
pest,
Samba Masuri.
infesting more
So
than
at that
80
point of
crops,
time, we were not
host
very
crops,
sure how many backcross generations
majorly
we had to
maize,
go. We went up to
sorghum,
BC4
and
generation,
rice.
and we deployed
And we
markers
know
not
that
only
sorghum
for selection
plays
of the
an
target
important
genes
role
called as
in
foreground
food and
selection markers. We also
fodder
deployed a
system,
set of SSRs,
especially in
which could
semi-arid
distinguish
tropics
between Samba
regions
Masuri and the donor
across
pest
Asia and Africa.
and Samba Masuri,
So this
with regard
pest is
to,
causing yield
the
losses up
presence or
to
absence of
30
the
to
genes.
80%.
Through this effort,
And
we
currently
could develop,
chemical control
one of
is
the
the
first products. Of course,
it
there
is
is
in
another product
management,
parallelly developed
but
called
it is
improved,
ineffective
Pusa
as fall
Basmati
armyworm feeds inside
improved
the
Samba
rows.
Masuri has got
And
three major resistant genes,
this pest
Exit,
also has
21,
developed
5 and 13.
resistance to many of
And,
the
this
insecticides.
is a success story of
So
not
host
only
plant resistance
of
is the best
deploying the right
alternative
kind of right
approach.
science for improvement, but also
So
about
with this background, we have framed our objective
collaboration with
is to
CCM is
identify
continuing for the
the
last
molecular
almost
basis of
25 years or
resistance to
28
this
years.
pest.
And
We
this variety
have
is doing
taken
really well.
242
And what is
minicore
important for you
sorghum
to
accession
understand is
from
that
ICRISAT,
under field
and
conditions,
this
it is even
minicore
as on today, it is standing
panel
very, very tall in
represents
terms
the
of
global
resistance.
diversity.
As you can see here,
It
on
helps
the
in
right side, you have got
identification of rare
panel which has
alleles
got improved Samba
as well
Masuri.
as
It is totally
novel
free from the
genes.
disease. Already grain maturity has started. On the
So
left
this
side,
is our
Samba Masuri is there.
we
wherein
framed in
it's severely
multi-step
affected by bacterial blight
screening,
disease. And,
including a
this,
field as
has
well as
already been
laboratory.
described by my colleague,
So in
Dr.
field we
Srinivas
have
Prasad,
screened under
wherein the background
natural
of improved Samba
infestation
Masuri, another
condition as
gene
well
called
as
Exit38 was added.
in artificial
And, this
infestation
also Dr.
condition
Srinivas Prasad has already
using
narrated.
a screenhouse
And another
facility
interesting
available at
development is
ICRISAT
a recent release
for
called
the,
as
across
DRR-85,
2
which
seasons.
combines
Here
bacterial
the
blight
observations
resistance
we
with
have
gallage
taken
resistance.
are
It showed immune
like
level of
foliar
reaction
damage
in
score, glossiness,
several
vigor,
gallage
and leaf
hotspots
sheath
along with bacterial
pigmentation
blight resistance.
from the scale
And,
1 to
we
9
have added a
Davis
trait of
scale for
salinity
the
tolerance,
foliar
the
damage
background of
score,
improved Samba
and
Masuri, and developed
other
DRR
traits
than
1 to 5
58.
scale.
Then another important
And
development is
under artificial
adding low
also
soil
foliar
phosphorus
damage
tolerance,
scale
which,
for 1 to
can
9
reduce
scale.
phosphorus
And
fertilizers.
to
It is
take
doing really
the
well in
account of
Gangavati area of
antibiosis
Karnataka.
mechanism,
And,
we have also
finally,
conducted
one
detached
important
leaf
version is
bioassay
development
for
of bacterial
the same
blight,
panel
blast, and then
242
submergence tolerance
mini core
version.
collection.
This
And
was
we have
recently released,
here
wherein
we
Dr.
have
Fiaz has
taken
taken a
leaf
lead
sample
role
and
in
kept
this
in agar
development along
medium,
with Dr. Prasad.
and we have
And,
infested the 10
we
neonate
also
larvae,
have,
and after 5 days we have
a
taken
success story wherein we
observation for
transferred,
the leaf
QTL
damage
with high
score,
zinc and
then
developed a
number
version
of
improved Samba
larvae
Masuri called
survived,
DRR-48.
and the
And
weight of
finally,
the
this
survived
is a flagship
larvae.
product in
So
which
from
it has
this
got
we have
multiple
taken
traits.
bluff
It has
values,
got,
and
bacterial
we
blight
taken
resistance
further
controlled by
the
21, 13,
genomoid
and 5,
association
blast
study.
resistance controlled by PA54, and it also has got
that is
yield
all about
enhancing genes,
phenotyping,
SCM2
and
then OSSPL14.
genotyping we
It's a
have
collaborative work
GBS
wherein we
data
got the
already
donor lines from
available
IRRI,
at
and
ICRISAT.
this variety
So
is doing really well.
we
In
have
fact, this
aligned
Kharif
that
season we had
GBS
a Darna
data
Bay
to
farmers
sorghum
wanted more
reference
quantity of
genome,
seeds of
and
this
we have
variety,
got 1 lakh
and it's spreading like a wildfire in some of the growing
73,000
areas.
SNPs, and after
And,
filtering
this is what
the
I would like
minor
to tell.
allele frequency,
through
we have
proper
got
planning and through proper
1,70,000
deployment,
high quality
we could add value
SNPs.
to Samba
So
Masuri
after
by
that,
initially
we
developing
have
bacterial blight resistant versions,
corrected
then blast
with the
resistant versions,
population
then BPH
structure.
resistant versions,
Then
garment resistant versions,
did
and
Jivas
versions which have got
at
tolerance
using
to abiotic
GAPIT
stresses, and finally versions of Samba Masuri which
package
can
in
give
R
higher yield also.
using a multi-locus model
with
like
better,
models like Blink,
nutritional
Farm
profile.
CPU,
And,
MLMM
we could do
at
the
a
same work in
significant
Swarna's background
threshold
also,
of
wherein along with,
2.9
bacterial blight
into
and
10 to the
blast
power
resistance,
of
we could transfer
minus
some of the yield
7.
enhancing genes
So
also.
after that,
And,
the
I would
resultant
like to talk about
SNPs
only
we
about-
have
just I'll take half a minute,
passed
sir, in
with
this.
this
Some new
GIF
tools are
annotation
available with regard to
file
accelerating
taken from
breeding for
NCBI
durable biotic
version
stress
3.62.
The first and
Then
foremost tool
after
is
that,
gene
we have
editing, wherein a
checked
group,
for
collaborative group
genes
led by
present
IDRI,
within
along
plus or
with,
minus 50 kb
Heinrich
at the
Heinz,
region.
University in Germany.
So
They
coming
have
to the results,
edited at
all
least
the
3 or
traits
4
showed
loci.
significant genotypic
These are genes
variation
called
with
as,
more than 0.8
susceptibility genes
heritability
like
for leaf
XA7,
sheath pigmentation
XA13
and
other,
moderate heritability for
XA genes which are known
glossiness
to be
and others
recessive genes.
traits
So the
with
promoter
low
part of these
heritability.
genes
And
are binding sites for
when
transcription
coming to
activators
the
called
correlation
TAL
between the
effectors. They
traits,
could edit the promoter part of these genes.
trait
And now we have
vigor,
got
glossiness
durable
and
resistance against
foliar
multiple
damage
strains
score
of
are
this particular disease.
they are
But,
moderate,
developments are
moderately
parallelly going on in India, identifying
associated.
different targets.
But here
But this
leaf
one
sheath
example
pigmentation
wherein,
we got
we are
weak
expecting the lines to be
correlation. But
received in
earlier
India, and then
studies
we'll be
showed
testing
that
against
leaf
different
sheath
strains in the
pigmentation
country.
is
And
contributing
another
resistance
important tool
to
is
pest.
called as haplotype-based breeding.
And recently
You all
in
may be knowing that
sorghum also
haplotype is a group
the
of
flavonoids
DNA
are
markers
shown
which are
detrimental
closely
to
linked
the
or
fall
co-inherited.
myome.
And we have got several
as they
favorable
belong to
haplotypes and then several
ordinal data, so
non-favorable or
we did
unfavorable
corresponding
haplotypes for
correspondence
several
analysis
traits.
where we
For
converted
example,
the
for
scores
yield,
into
these are all the
resistant
different genes
classes
and
for
their
both
favorable
the trait
haplotypes.
leaf damage
Deploying
rating
this approach,
and
my colleague
leaf
Dr.
sheath
Jyoti has
pigmentation.
developed
So
a
after
very good
association
variety
we
called
got
DRR-87 in the
certain
background of a
classes like
variety
partial
called,
resistance and susceptible classes
Swarna.
were
And this particular
strong,
breeding approach we
they
followed
showed
a
a strong
recombination breeding approach
association.
with a reduced backcross breeding approach.
So
And
after
this
the
version has got
GWAS, we
tolerance
have
or
got
resistance
total
against multiple
45
pest and
significant
diseases.
marker
And
trait
of course, speed breeding
association
is something
for
which
the
can
4
really
traits: foliar
accelerate our
damage
efforts
score,
in
leaf
terms of
damage
rapidly
rating,
developing,
leaf sheath pigmentation, and
multiple
larval
biotic
weight.
stress-resistant varieties
And some
as
of the
compared to
traits,
just 1 or 2 seasons
you
in a
can
year.
see leaf sheath
With collaboration with
pigmentation
IRRI,
and
we have
larval weight,
demonstrated that
as
up
well
to
as
6 seasons can be taken
LDR
up.
showed
And even
more,
in wild rice also it
they
can
occurred
accelerate
in
the
more
breeding
than
process.
one models.
And finally,
these are
one
the
important
total
component I would like to touch upon
significant
is
MTAs.
artificial
And
intelligence.
through
If gainfully
Blink
deployed,
model, we have
it
got
can be used
10
in terms
significant
of
MTAs, where
predictive risk
you can
modeling,
see the p-value
A
ranging
powered
from 10 to
phenotyping,
the power minus
and it can accelerate
7
the
to
process
10
of
to the
genomic
power
selection or
minus
genomic
100.
prediction.
And it
even
also
in gene
showed
discovery
good
also it has got its own
phenotypic, they explained a good
use.
phenotypic variance
And
ranging
speed
from 3
breeding
to
can, be
66%
accelerated through the
of
application
the
of AI
variance.
tools.
And
If anybody is interested, I was
so
deeply
you
impressed
can see
by
the
reading this
Manhattan
paper
and
by Farooq
respective
et al.
QQ
It
plots
gives a very
of
good
the
workflow
association,
regarding how we
the
can deploy
foliar damage
artificial
score,
intelligence in breeding process.
one for one
And finally, just in
SNP
a slide
and
or two, I'd
4
like to tell
significant
what did I
SNP for
gather in the
leaf sheath
last several
pigmentation,
years of my
and 2
work at
are on the
IRR.
same
So
6th
if you look into
chromosome.
it, breeding
And
climate resilient varieties
one
through
for
genomic-assisted breeding should be our
and
target.
4 for
Towards this,
survived
we
larva weight,
clearly demonstrated
where
that
2 are on
interinstitutional
the first
collaboration, not only
chromosome.
between IRR and CCMB,
So after
IRR
the
and several
annotation,
other
we got
institutes, whatever I
know
presented is
the
because of collaboration, not only within the
genes,
institutes, between
candidate
different
genes
disciplines,
present with,
but also with
within
other
plus or minus 50
So I would like to
kb,
mention here that
where we
interinstitutional
got
collaboration
this
is the key for success.
description of
And
the
another important point is that
identified candidate genes.
molecular breeding
Example,
is already
FBB
started
for
delivering products.
the
Already
trait
about 2
LDR,
million or 2.5
and then
million hectares is under
other
molecular
proteins
breeding
for the
derived
leaf
products
sheath
in the country, in
pigmentation
rice
like
particularly.
kinase,
And another
and
take-home
as well
point
as
is that
EBS domain.
you should not
And
target
then
improving only
this
1 or 2 traits. You should
larval
always
weight
be
actually
ambitious or
showed a
over-ambitious,
good
and multi-trait packaging through
related
genomic
proteins
assisted
like
breeding
proline
is a way for us to
dehydrogenase
forward.
and
We have
pectin
demonstrated in the
acetylase
background of Swarna and Samba Masuri, and
as well as
CRRI has done it with
peroxisomal
in several backgrounds.
nicotinamide.
IRA has done it with the help of
And
NAPV
also for
in terms of
LOH2
adding value to several
related to
basmati
lipids
varieties.
and
finally, what I
related to
would like to
calcium
mention here is
flux
that
and
genomics should be
glycosyl transferase,
increasingly
as well
deployed
as
not only for gene discovery
serine
but also
or
for
threonine
accelerated
specific
gene
proteins.
deployment. And what is
So
important
previous
for
studies
as a
also
country
have
is that
reported
there are several
some of the
novel
proteins
sources
like
of resistance. We should work towards
peroxisomal
identifying
nicotinamide.
the functional
They
basis
play
of
important
resistance
role
or
in
tolerance.
jasminic
And
acid,
we should
other oxidative stress
work
related
more in this
pathways.
direction, just like the way
Actually
Dr. T.R.
through
Sharma's group has
flavonoid
worked.
based
And of course, the new tools
host
which I
defense.
described, They can certainly complement the process of
and
breeding.
calcium binding
But
family
finally,
proteins.
I would like to mention here is that
calcium
we
flux
started
will
our work like a bullet
help
cart.
in
We were using
signaling,
RAPDs, ISSRs
and also
for
the
breeding for
other
biotic stress
proteins like
resistance. When
pectin,
I joined,
acetylase,
I
and
started a program for
lipid-related yellow
marker-rested breeding, and
yellow
we were
H2,
using a few
and
markers
they are
like
involved in
SSRs,
cell wall
ISSRs, CAPs and all.
remodeling, and
now
other
my
things
team
like
is traveling in
glycosylation
an
transferase
SUV wherein we are
involved in
deploying high-throughput
salicylic
genomic
acid
tools,
mediated
not only for
signaling.
gene identification but also for gene
So in
deployment.
conclusion, our study GWAS
Finally,
on
this is my concluding
242
slide.
sorghum mini-core collection, we have
A breeder can
identified
ask a question,
45
whether can he use
significant marker trait
biotechnological tools
associations
for
for the
improvement of a
fallernium
trait?
resistance,
The
and
answer is
through
definitely
link
yes, but we need to remember that
MTAs
biotechnology is
for
one
the 4
among
traits,
the several tools
explaining
available
3
for a
to
breeder.
66% of
At
the
the end of the day,
phenotypic variance,
biotechnological tools like MAS,
supporting the
molecular breeding
involvement of
genomics,
multiple
can only
loci
complement
from
and then
small to
supplement
large
the process of
effects,
breeding.
and
It will never replace the process
annotations
of breeding. And we should
showed
embrace
that
all these
they are
modern tools
involved,
in day-to-day
those
breeding.
candidate genes are
With this,
involved
I would like to
in some
thank all my
signaling,
colleagues
redox
who work with me.
regulation,
Some of them are here
flavonoid
also, and also
biosynthesis,
my
as
funding
well as
agencies
cell
who
wall
have been very, very
modification.
extraordinarily
So
generous
in
in terms
future,
of
the
funding all these
validation
efforts.
of the
Thank you, one and all.
identified
Thank you, sir.
candidate genes are needed than using
Thank you very much,
transcriptomics
Dr.
and metabolomics
Sundram.
to understand the
Very nicely
mechanism of
presented
resistance,
how we can
and then
map
next the development
new
of
genes for
markers for
different
these
traits like
candidates,
blast,
and next
BLG,
further
and also insect
involvement of
resistance,
them in
and then how to
marker-assisted
deploy.
breeding.
Actually,
Thank
I
you.
think major impetus on molecular
Thank you,
breeding
Ms.
was
Ranjita,
started
for
from Indian Institute
sharing
of Rice
outstanding work,
Research,
and
where
also
you are
two
heading now.
demonstration of
And
some of
almost all
the tools what
varieties
our
which we
earlier
are
presenters
cultivating
presented,
in India
both
are
in analytics and
marker-assisted
also molecular
variety
tools.
in
Thank you for
rice.
that.
So
Overall.
that is
And
all
now I
because
requested
of
Dr. T.S. Sharma to
leadership which you
share
are
some
providing there to
conclusion.
this whole rice improvement program in the
Thank you,
country.
Dr. Gowda.
Since
So,
we left with
in this
very last
particular
time,
session,
still
we
we can
had
have
1 question.
8 presentations, including
So it's
3
difficult to
oral
choose
presentation by
Dr.
our
Tikku
young
and
colleagues.
yourself. You can decide yourself.
And you all will agree with me that this was one of the most
चलो आप पूछ लो पहले.
enriching and knowledgeable session which we had on rice. And I would like to say
The molecular breeding of
4 to 5 points
Yeah,
on
I
this
couldn't hear
particular
you, so can you
sessions.
keep the mic
Particularly, we learned about disease and insect
we have
pest
seen in majority
scenario
of the crops,
rice
including rice,
in the
that
country,
the developments what are
their
happening with the
prevalence,
molecular breeding are with
significance,
respect to the
and
stable
management
varieties. For that
practices.
matter, Samba Masuri,
Then a
which is a
success
well-known
story
variety from
of
almost all years.
discovery and
Now
deployment
you
of
suggest your opinion
blast
I want to
resistance
know.
gene
Whether we need
PI54
to
and
start
its
a fresh
impact
breeding
on
with like
rice
magic
breeding.
populations involving all
And
these
then
genes,
Sir,
or
mapping
we have to
and
go with the
characterization
stable
of
variety,
genes
means
for
ABT
various biotic
advanced
stresses
varieties
and
should be
their
taken, my opinion.
introgression in
ABT
rice
advanced
breeding
varieties should be taken
using marker-assisted
into this,
selection and
molecular
also
tool introgression program.
that's
successful
one way to
examples
that's one way to
of,
look at it.
mass-derived
We always,
products
when we started our
which we
molecular
have in
breeding
the
program,
country
we look
now.
at low-hanging fruits.
And then
one easy
challenges and
strategy is
opportunities
marker-rested backcross
of rice
breeding.
improvement
But believe me,
globally. And
in forward
available
breeding, if
new
you deploy the
strategies
right
with
set
the
of markers,
breeders.
the right set of traits,
And
we
finally,
can
we
transform
had 3
breeding.
presentations
One typical
by
example is DRR
young
Dan
colleagues
75.
showing
It was
their
not
work,
developed
and
through
I would like
backcross
to
breeding. There was only one backcross with some
all the
include Samba
speakers,
Masuri,
particularly
and
our
further
young
it involved
speakers
crossing with several
for
parents.
their
But
nice presentation.
we have been successful
And I
because
would
we had a
also like
large
to thank
population
my
size. We were
co-chair,
able to
Dr.
identify a plant
Viresh Gowda,
which was
head
similar in grain
R&D
quality to
via
Samba Masuri,
Biosciences,
and we could
and
transfer at
to
least
report here
4 or 5 different
Dr.
traits. And another example is
Suma
DRR Dan
and
79,
also
wherein
Dr.
at least 7 or
Gurudutta.
8 traits have been
Besides
added in the
that,
background. And that
thanks
was
are
both
to the
DRR Dan
organizers
75 and
for nicely conducting
DRR
these
Dan
particular
79.
sessions
They were not
and
released
giving us
through
opportunity to
NAL
be
trials.
here
They
this
were
morning.
released
Thank
through
you very
ordinary
much.
trials, wherein the lines are to be tested against the best variety and tested for 3 years. So I think
Thank you so much, sir.
while
Now we take the privilege
could
to
be a
extend
case
our sincere
wherein,
gratitude to our
good
learned
variety,
speakers.
we need to make
I
it
call
great
upon
by
Dr.
adding a
M.S.
few
Prasad,
traits.
sir,
I think we should
Dr. R.M.
invest
Sundaram,
more
sir,
on
and Dr. Amresh Tandal,
approaches
sir.
like forward
Please
marker-rested
grace
forward breeding
the
and
dais,
then
and I
recurrent
request
selection
our Honorable
and
Vice
magic
Chancellor, Dr.
population,
P.L.
and
Patil,
invest
sir,
less on
to do the honors.
marker-rested backcross breeding, sir. That's the hard lesson I have learnt.
And I request
From student,
chair and
any
co-chair also to join the felicitation of our learned speakers.
Yes, go ahead, sir.
So, okay.
Good afternoon, sir.
yeah.
Myself, Harish, PhD scholar, sir. Sir, in GWAS, we are identifying SNPs for particular traits, sir. So now there is pan-genome approaches are available, sir, where whole set of genes are identifying, and we can do large introgression, sir. So can we use some phenotype and genotype matrix of pan-genomes for GWAS analysis as an input?
You have raised the right point. Initially, our approach in GWAS was only using the primary gene pool, that is the varieties and land races. But of late, the thinking process has moved differently. And we have started deploying even some of the wild relatives and other exotic genomes also. But you need to understand that genome-wide selection or genomic selection is a work in progress, because we have just started with our ACRIP trial data, both phenotype and genotype data. Every year around, we have got the phenotype data for at least 12 or 13
now further
different
we take the
traits
pleasure
every
of
year. But it's a work in
felicitating
progress.
our respected chair and co-chair also on behalf
we
of
should
University
certainly
of
move
Agriculture
from
Sciences,
the primary
Dharwad.
gene pool to a pan-genomic approach. You are right in that. But progress will come in, say, another 3 or 4 years or 5 years, when we'll have a
to
large set
Dr.
of products developed through genomic selection.
Videsh Gowda, sir, who is a proud alumnus of this prestigious University of Agriculture
Thank you,
Sciences,
Dr.
Dharwad,
Nice
who did
presentation.
his masters in this
Now I
university,
would like to invite Dr. Amresh Chandel,
and to chair now
Rice
Dr.
breeder at
T.R.
Monsanto,
Sharma, sir,
Hyderabad.
former Deputy Director General, ICAR, New Delhi.
Respected Chair, Co-Chair, honorable dignitaries, and all the
Thank you,
delegates
sir.
present here.
And we also extend our sincere thanks
Thank
to
you for this
both the
invitation.
rapporteurs, Dr. Suma Biradar Madam
We
and
are
Dr.
all very
Gurudat
proud
Hegde,
of
sir.
the fact that
And we
India
request
has
them to
achieved
hand over the
number
proceeding
one
of the session by tomorrow
status
morning.
in rice production.
Thank you, sir. Now we have a
And all
very
my
special
fellow
event
colleagues,
of exchange
scientists,
of
distinguished
MOU
scientists who
between
spoke
University
before
of
me
Agricultural Sciences, Dharwad,
have a very big role
and
in that.
private maize company.
And
I
also
request
all of you have
Director
very
of
big role
Research, US
who have
Dharwad, to
contributed
join
to
us
this
on the
success.
stage.
But this conference
And also
on
the
multiple disease
representative from the
resistance is a
company.
very apt
And I
moment
request
where
Honorable Vice Chancellor,
we know
sir, to
that
speak
we
few
have a
words
lot
on
of
this occasion.
potential in rice
Thank you,
and
Dr.
how we can go
MD Patil.
forward
So
together as a private
you
sector
can
and
take
public
this is a
sector.
commercial
Good
break
afternoon.
or
My name is
commercial
Amresh
opportunity.
Chandel and I work in
So we
Bayer Crop Science.
Want to commercialize
I will be
our,
presenting a private sector
technologies
overview
released by
of
our
how
own scientists.
we can work together
Maybe varieties,
and what are the
hybrids,
new approaches
and
we can
also
take
the important
in
inputs.
achieving the
So kindly
success for
refer
rice
to-
production
I
in
request
India
all the
and also in
industry
Southeast Asia.
partners, please refer to Agri Innovate
So my
India
presentation
Limited
is
website.
Overview
We
here,
are commercializing
context,
all our
then modern molecular
technologies
tools, gene
through Agri
pyramiding
Innovate
strategies
India
and
Limited,
regional evidence,
and choose the
CRISPR
technology
advances
required
on multi-stress
and express your
regulation,
interest, and
digital phenomics,
through the material transfer
economic and
agreement,
sustainability
we will
impact for
have
farmers,
the commercialization
and
of our
also buyers'
technologies.
arise journey and future outlook.
And
Then the
we have
conclusion.
already
I know the time is
commercialized
running
2 color cotton
late.
hybrids.
I will try to
maize
finish
hybrid,
fast because we
and,
have some interesting oral
biofertilizers.
presentations also.
So like this, the list is go on.
Now,
And
what are the
also we
field
have several
realities driving the modern
value-added
rice
products
breeding?
from College of Community Science. They are also
These are
available
the key
for
challenges we
commercialization
see in the
for,
field.
the young
Insect pests,
entrepreneurs
we
or
all know.
ladies to take up the
Viral
activities.
diseases, which are becoming
So
very important now.
here,
Multiple pathogen
today,
pressures and rapid
so we are
biotype shifts are happening.
having MOU between one industry.
In the conventional
which is
breeding,
that,
we do have constraints like difficult to stack multiple resistant genes without introducing linkage. Long breeding cycles
Gen
delay
Z seeds
delivery of
and
improved
US Dharwad
varieties.
for
Phenotyping
our Dharma
multiple
hybrid
stresses
and
simultaneously
another,
is an
the
inconsistent
line
and
in the
labor-intensive.
Another hybrid in the
Limited ability
pipeline.
to track complex
So
gene
Dharma
interactions
hybrid,
using traditional tools.
the features will be indicated by
Now,
our
why precision,
maize
speed, and data
scientists.
integration matter?
So both are US Dharwad
Because we need
products.
to combine many resistant
Each are
traits
sold
cleanly and
for
efficiently.
3.6 lakhs, I think.
Faster
3.3 or
selection cycles
3.6
enable
lakhs.
quicker
So in
deployment
addition
of
to Agri
resilient
Innovate India,
products.
so you can also consult
Data-driven
Directorate
phenomics
of
and
Research,
genomics
US
improve
Dharwad
accuracy,
for
confidence, and
commercialization.
decision
Thank you very
making.
much.
And integrating molecular tools with digital analytics accelerates genetic
Just
gain
to
under
brief you
multi-stress
the
environments.
variety details, and with the
What
support
are our
of our Vice
advanced tools
Chancellor
and platforms for rice
successful
resistance
licensing
breeding?
of our PPMC, head Dr.
We
C.R.
do have
Patil,
molecular
sir,
tools
we
like
organized
marker
one
assisted
maize
selection,
hybrid
which
field
all
day
of you are very
during
much
September.
aware.
There are around 15 private partners
Then we have gene
participated,
silencing, RNAi
and
approach.
they shown the interest in some of the hybrids. And this is
genome
one
editing,
fruit
CRISPR-Cas,
we are getting out
which
of
is
that
a
one.
new
And there are so many
upcoming
people,
technology.
hybrid,
On the
they are
advanced
asking
platforms,
for
we have
to
speed
evaluate
breeding,
their,
which
in their field.
is
And
changing
they
the
may
way
come.
we are
They have shown interest in
getting
next
genetic advance
course of
faster.
season. They will come and to have MOU.
Then double
so
haploidy.
that the fruit
Another
of our
approach.
scientist will go to
And
end user,
genomic
that is a
enabled trait
farmer.
discovery.
These are the effort of our two breeders, Dr. S.C. Talekar and Dr.
This is how
Kachapur.
integrated pipeline looks like for multi-pest and disease resistance in rice. First is a discovery phase, then we do markers and edits, then the stack the design, rapid cycling using speed breeding, phenomics, and it goes to the field testing. Then predictive analytics are applied, and finally deployment and impact happen. This all can be done within a span of 5 years now, but previously it took 12 to 15 years. So this is how we have to integrate our multiple disease resistance
To move
in
further,
rice.
now we have a panel discussion
And on the genome
on
editing
this
strategies
theme
for
of
disease,
on the crop rice.
this is the way
I
it
request
goes
Dr.
as
R.M.
gene
Sundaram,
knockout,
sir, Director
optimize the
ICR
alleles,
IRR
promoter
Hyderabad,
and
to
cis
chair this
editing,
panel discussion.
multi-target stacks,
And then
and
I
again
call
validation
upon
and deployment in the
Dr.
markets
N.G.
where we want to release the
Ramachandra,
product.
lead breeder, rice breeding bioseeds Hyderabad.
On gene pyramiding,
Dr.
context
Venkatesh
and rationale
Kulkarni,
in
Director
South and
R&D,
Southeast
North
Asia.
Biogene Seeds. Mr.
Why do we
Devaraja
need gene
Achar,
pyramiding?
Head Biotechnology, Indo-American Hybrid
Because we have
Seeds,
major
India
regional
Private
threats
Limited,
like bacterial
Bengaluru.
leaf blight.
Dr.
It is very
Chandrasekhar
diverse,
S.,
evolving
Rice India
races
Breeding Lead,
across India,
Kotiva,
Bangladesh, Vietnam,
Hyderabad.
Philippines.
Dr. S.K.
Rice blast
Prashanti,
again,
Professor and Head
multiple,
Biotechnology, US Harvard,
Magmacorte
and
races
Dr.
are there.
Vinesh Gowda,
BPH
Head Asia
has
Rice
different
Breeding,
biotypes
Bayer Crop
and
Sciences,
emerging
Hyderabad,
bacterial
to
leaf
join as a panelist.
streak. BLS is becoming very important, especially in Southeast Asia. Our production goal is to enhance durable broad spectrum resistance without compromising the yield or grain quality. What are the breeding strategies which we employ? First is a marker-assisted pyramiding, which remains the most effective and scalable approach. There have been recent evidence where ERI, Bangladesh Rice Research Institute, and many Indian programs have effectively utilized it and released the products. Optimal pyramids include 3 to 5 resistant genes or QTLs to avoid linkage drag or yield penalty, and increasing use of newer R genes like XA23, 47, 50 in the elite backgrounds. And what is the target profile we are looking at? A high yield product under Swarna or MTU 1010 mega varieties background, suited to irrigated and rain-fed lowlands, and strong tolerance across multiple pathotypes.
Good morning to you all.
Which is very evident from the ECRIP trials, where
we are here,
both public and
for,
private partners give
important,
the
panel
products for
discussion
testing,
on
and
the
we are
theme,
seeing
particularly
very good
breeding
products coming
crop
out of that.
varieties for durable resistance to multiple biotic
Gene pyramiding
stresses.
recommendations from
And
my
in this
side
particular
here,
panel discussion,
based on evidence, the first recommendation is, I'm sure many of you might be following this, but this is just to reiterate
we have to
that broad
address increasing
BLB plus
complexity
blast
of pest and
resistance is the
disease
most
pressure.
validated,
And
where XA 5,
we also
13,
need to
21, and
discuss about the various
PI54,
strategies
PI9,
in
these
terms of
combinations are
rapid or quick
proven
development of
durability,
better breeding
where
lines,
many
which can
products have been released.
tackle multiple
The second
biotic
can
stresses,
be on XA
and how we
23,
can integrate
21, 5,
conventional breeding
and PI54,
with
or
molecular tools,
XA 47,
AA,
21,
and
and
also phenomic
PI9
tools, and
for the
how
enhanced
to bridge the gap
durability.
between
And the
research,
third is a
varietal release, and then
multipest where
farmers' adoption.
BLB, BPH, and
There are a
BLAST,
few
all
questions
three
I would like
can
to
be
ask the
combined in one
panelists. I'll
product.
just throw a few questions.
And the fourth will
we
be
can have
advanced
about,
next generation options where
3-minute,
we can use CRISPR strategies
submission
also.
by each panelist, following which we'll have a round of short
What are the
round of
practical
discussion with
deployment
the,
and expected
audience
outcomes?
Also. So the broad questions which the
On the
panelists
breeding
can
methods,
answer in addition to
marker-assisted
telling their own
selection
views
takes
is
2 to
that
4 years for
my first
integration,
question is,
where
are we
you
all
use
relying
type
on major
markers
genes?
for major genes and validate
is there
with
any
background
scope or
recovery
limitation of
phenotypic
deploying
screening, which
the
you all
minor genes
very
or
well aware.
QTLs in a breeding program?
Outcome is 20 to
How do we
40% lower
predict
disease
resistance
severity,
breakdown before it
maintain or
actually
improve
happens?
yields,
Are there any
and stronger
strategies?
resilience against the
And the third
evolving
question is,
races.
can artificial intelligence and
So
machine
this will
learning
help us in
reduce breeding
regenerative and
cycle
climate smart
time
rice
meaningfully?
systems.
The fourth
Recommended
question
target
is,
backgrounds
what is
are
the
again
ideal
the
gene
megavarieties,
combination, particularly for
MTU1010,
Indian ecosystems?
IR64,
Are we there, for example, for
CHR and
bacterial blight, I can say with
PSB
confidence.
series,
And similarly, based on the presentation given
and
by
partners and
Dr.
pipelines. So we
Srinivas
have to
Prasad
work
and Dr.
together.
T.R. Sharma.
As Dr.
We are
Sundaram
also
told
there
that
with regard to
there is a
blast
very good
resistance,
linkage
but
between
with regard to the
partnership
other
between
insect
national
pests and
programs and
diseases
state
in different
programs.
crops.
I would add a
Can we
private
tell
partner
what is the
also
ideal
there.
gene
So,
combination depending on
come up
what
with the
particular
projects where
ecosystem in which you are
public-private
growing?
partnerships
And
can
my
also
final
work
question
together
is,
in a
how
NARS
do we ensure
program,
pyramidal
which will
genes,
help to fasten
how
the
can they
process.
remain effective post-release? Or how to prolong
On the CRISPR, let's
life
see
of
what are the rapid
pyramidal
progress
gene products? These
and
are
why
all
the CRISPR
broad
matters.
questions which the panelists
Because
can
this is a
attend, but
precise
I would like
targeted
each one of
edits
them
with
to,
no foreign DNA.
submit their
Faster breeding cycles, minimal yield penalty,
views or,
and
try to
ideal
answer
for
at least one
South
of these
Asia and
questions.
Southeast Asia.
I now invite from my
Which key traits
right
are
side,
improved?
Dr. Veeresh
Yield and
Godaji.
plant
We have
architecture,
about 3 minutes to give
Sure.
submission.
higher yield,
Thank you, Dr.
unchanged
Sundaram.
quality,
So
deep
my
free
viewpoints,
knockouts,
especially
give
you know,
22 to
right kind of a
38%
gene
BLB
combination
lesion
or right
reduction.
approach to tackle some of these challenges
And
is
also
it's always
OS,
what
APL,
customer
GL,
needs.
W7,
First, we need to
GL,
think from
edits,
the farmer what
improve the
farmer
grain size,
is actually
nutrient flow,
facing a problem.
grain quality and
And it
nutrition
should be
is
not
also improved.
across India or across overall
For the
rice,
BLB blast and
community we want to
multi-stress
solve.
resistance,
It is always
also
essential
CRISPR
to
has
know
been
that
utilized.
what product and what product
These are
concepts
the
we
major
wanted to build.
breakthroughs where
Based on
broad
that, I think
spectrum
if we
BLB
can really
resistance
design a right
effective
gene
against diverse
combinations,
Asian and African
looking at
strains
the
was
challenges what
demonstrated
customers are facing,
in
maybe that
IR64,
will
Sierra,
help us to
Sub1 and other mega
address some of the
varieties.
challenges.
And there was
on the
no
top,
major agronomical
yield is always
penalties.
important. Every time without yield,
Additional disease
any,
resistance targets
upgradation we do on the particular
where
product, it will
this
be not
B9
completely
mutation can
accepted by
enhance
customer, because customer
BLB
always
resistance,
or a farmer always
BLB
wants
edits,
improvement
and
over
both
what he is
BLB
doing
to
today.
enhance
Not only just
architecture
addressing the
benefits and
challenges, it
multi-disease
is also
packages.
essential to have yield improvement. That's
For
why I can
abiotic
say
stress
a lot
also,
of now
drought,
new technologies which are available,
UV tolerance, arsenic
improving
reduction,
yield
and
along
other
with bringing a
enhancements
lot
like
of this
herbicide resistance
the tolerance. It
and
can,
grain
it will be a
appearance
right
can be done
approach,
using
I can say. And
this
most of the
technology.
genomics what we learned today.
What are the
if
regulatory
we can look at
status and
the animal
application in
genomics, I
Southeast
think a
Asia?
lot of advancement happened even before the plant.
India is the
So
fastest
maybe it's
deployment.
a good
You all
approach
know,
to look at what
last year
advancements
we
are happening
released
in
Pusa
animal
rice
genomics,
TST1.
which will actually help us to bring a
DRR
lot of
Dhan 100
quantitative
Kamala.
approach
These are the two
to
gene-edited
tackle a lot of
varieties.
these major and minor genes what we are looking at.
On the regulatory
So,
part, SDN1 and
always we need to
SDN2
keep
edits
open
exempt
high
from
and
GMO rules
thinking
under
process
India
to
EPA
look at
Rule
some of the advancements so that
7-1
we can really
and
tackle
expanded field
site-specific
trials
or
after 2024
target specific
regulatory relaxation
problems we can bring it for
and
the
high
customer.
relevance to India, BLB
Thank you,
drought and
sir.
salinity.
I now request Dr.
In
Devaraja
Southeast
Acharya,
Asia, there is an ongoing
from
EV
Endowment
and national
Hybrid Seeds to give his
collaborations, testing of CRISPR
perspective
suite edited
So I would
BLB resistant
be,
lines in IR64
discussing on,
background.
marker-assisted breeding.
And commercial
It is one of
approvals
the
are slow, but
important,
edits without
key
transgene may
aspects for
be
any kind of a trait
non-GMO
improvement
depending
or
on the
development
national
of a new
policy.
varieties, in fact. As Sundaram sir said, from
Then
car to
integration
SUV.
with the traditional
In
breeding,
fact,
where
it was
CRISPR suite
if you see
promoters
the
are
advent
edits
of
or
the markers
complementary
in
pyramids
almost
like
20,
XA23,
2 decades,
21, PI54.
majority of the varieties have been
This
mega varieties
enables
have been
durable
developed and they have
resistance
been
without
commercially
relying only on the
viable
resistant
in the
genes.
market. So therefore, identification of key traits
These
is
are the
very
challenges.
important. But if you see the complexity of the
Off-target
trait,
editing
and the
reduced
evolution
in
of these
newer tools.
races, in fact,
So this
with respect to the
is
fungal
reduced
and
now.
bacterial races compared
Public
to the virus, in
acceptance
fact.
and IP constraints.
As
This
we
is
have
one of the
pyramiding,
biggest
the
challenge.
time taken what we are taking the
And
pyramiding
harmonization of the
genes or
regulatory
something like
framework.
that,
But
it
we
is
all are working
tenfold
together
higher
to-
than what the strains
to
or
overcome
the
these
pathogens
challenges.
are evolving.
I am
in
sure
fact.
within
To
few
combat
years
and
it will
cope
be
up to
overcome.
such kind of a things, it's very difficult, in fact. So
These
that's
are
the
some
reason
of the
where the
reports
gene
of
editing
pyramiding
approach
BLB
is one
blast
of
and
the
DPH in
new
elite
approach
backgrounds.
where you can
On
able
the
to
left
do it
side
in
you
short
see
of 2 years, where
marker
you can
assisted breeding
anticipate,
from
in
the-
fact.
from
And
the
now,
India.
not the developing or
Popular
incorporating the
cultivar
resistant
Tella
gene to the
Hamsa was
background.
improved by
Now you are
pyramiding
going to
these
edit the same plant
XA21
by
and
recessive
XA13 gene.
ways. That means either you are making the plant to be
And also in
not
Tamil Nadu,
recessive are resistant to for a particular
stacking
disease, in fact. So
of
therefore,
different
the
genes
fast
was
tracking
done
of those
in ASD
incorporation or
16 and
editing the
ADT
gene of
43.
the native genome is one of the
Similarly,
best
in
approach
China
than
and from Bangladesh,
putting
there
the
are some
donor gene
reports.
into this donor
These are very
genetics into this
latest
one because of the lot
development.
of linkage drag and that and this. Okay, of course
And
there are
multi-local
many approaches how come
validation,
genome association
evidence from the
or
trials.
other ways of these things.
Tela-Hamsa
But
pyramided
pyramiding
lines
the genes
validated at 3
in the
blast
native, in the
hotspots
major
and 2
genes are
BLB
fine. But if
locations.
you take the
They
QTL,
exhibited high
again, it's a
resistance
complex
across
with the genome
environment,
editing.
supporting the
So therefore, a
spray
lot of
reduction
issues
potential.
are there, but as a lot of marker-assisted breeding
Similarly,
is
BRRI
need to be
Dhan
fast-tracked
63 pyramided
with the
lines,
multiple
they achieved
tools, in
near
fact.
immune
The
scores
best
0
approach
to 1
is
across
presently the
tests,
gene editing approach,
and
which we
multiple
need
lines
to
demonstrated higher yield
take it forward
than the
for
elite check,
varietal
implying
development or
fewer
hybrid
fungicide
development in
and
rice.
bactericide applications for equivalent production.
Thank you very much. Now I request Dr. Venkatesh Kulkarni,
Again for
Director,
ASD 16 and
not
ADT
Biogene
43
Seeds.
with blast, BLB and sheath blight resistance.
Good afternoon, everybody. I am actually placed between two people
Farmer
who
impact.
both are helping us in especially rice
Yield and resilience.
marker
So
develop,
here we see
marker assisted selection
several lines in,
in
BRRI Dhan 63
in
having
rice.
more than 28 to 30
Whenever
gram per
my
hill
laboratory is
and near
overflowed,
immune disease
Dr.
scores.
Devraj Acharya helps
This
me
reduced
in
input cost
analyzing
and
our
lower the
sample
crop
and
loss
sending
risk.
the results to us.
And stacked packages
the way
in the
which
elite background,
the cost and the
both
speed
in China
that he
and India,
manages is
they maintain
very, very
economic
appreciable.
traits,
He
broaden the
has helped
resistance
us a lot
spectra
in developing our population, etc.
under the
The
biotech
second one
shift.
is Dr. Sundaram, because
And
in
again,
my
this is a
first
public-private
part of
workflows where
rice marker
we can
assisted
combine
selection,
resources and
honestly,
work
we
together.
suffered a lot because of the source.
Genomic resources for
the
BPH.
markers were in such a source that at the end of the day, product was not coming. Now,
Digital phenotyping and data-driven breeding decisions are now being taken
elite
across
sources
in
are
private
being
sector
impregnated
and
with the
public
markers, and
sector,
those are available for as a source
where
for
digital phenotyping
much
and
more
data-driven
validated sources are available for
driven breeding
development
decisions are
of,
very important.
especially blight and blast.
And then it combines
I have
analytics
some of the points that to be
and gives us
successful
a digital dashboard
in these
where we can
marker
take
assisted
decisions
selection
faster.
are the hotspots and as well as screening protocols. And
What are the
source of
sustainability
the material
benefits
is very important.
of resistant rice
These
varieties?
three things: a consistent
Outcomes.
management of hotspots.
First is a
and
fewer pesticide
pure culture of
application,
the organism,
lower pesticide
will
residues,
help us in
improved
getting
safety
the
of ecosystem,
fastest and accurate
enhanced
results.
resilience of climate
In addition
variability.
to that,
And for the
your,
farmer,
period
what is the
of,
value?
marker
Cost saving
analysis
from
and,
reduced spray,
before flowering, if the
higher
marker
and
data
more
is
stable
available,
yields,
then we can
reduced
really
production
manipulate
risk,
within the field and get
and better
the things.
access to the premium markets.
When we said that we are going for 4 markers, then we
So Bayer has a
means
rich
1 out
history
of
of
16 will
bringing
be our
disease-resistant
plant.
products
so
in the market.
Based on that, we
Bayer is the
increased the
first company
population
which,
and we got the
one
results
of the first company
that
which
actually
released the
helped us.
BLB
on
resistant
and whole,
hybrid,
in rice, it
BLB
is
and
working
DPH
very
resistant
well for
hybrid back in
marker selection
2008
to address
with
the
the name
bias
Arise Dhani
stresses.
DT.
And in addition to
This
the
is the
rice, in
hybrid for the
tomato, we
late
are
maturity
seeing a
market,
lot of success
and
on
it,
that.
it provides protection
Here,
against both BLB and BPH.
one of the challenges as a private sector
Later on,
person, we are
Arise
also working
644,
on
which is one of
hybrid
the
breeding.
best-selling hybrids
So
till date,
one
was
of the
also in
protocols
progress
that we have
with
developed is that
BLB
once
and-
we develop a
and
B
blast
line with the
genes,
marker,
and it is available in the market.
then integrating
And
it,
over the
making
years,
it
you
in A
can
line,
see we have been
we are reducing the
bringing
time span
products which
and,
are
we
climate
doing
resilient
some
and
very good
having
work
both
on that.
biotic and abiotic stress
However,
tolerance
a lot of our platform
across
is getting
multiple
shifted
segments
to
and markets.
TGMS line. There will not be any
Today,
big
at Bayer,
difficulties
we
because
are
it's as
giving
good as any
products for
pure line
all the segments,
development.
all the market segments
Durability is
present
question
in
is
India and
consistent.
Southeast Asia.
Even we go
And with
through the
these resistant
marker-assisted
genes.
selection. In the hotspot,
Arise
consistent
8433 DT,
screening
8455 DT,
would help us in
7006. All these
in deriving durable
products are
thing.
there.
Further, first few
This is the Arise portfolio
multiplication
where
of
climate
breeder
resilient
seed
performance
in the hotspot
across
itself
diverse
will
and
also help
variable
in
environments,
enhancing the durability of
abiotic
the
stress tolerance including
material
drought,
that we have
submergence and
developed.
lodging,
On AI,
biotic
recently,
resistance package against
our
major
team
pest
got-
and disease
our
like
R&D
BPH,
team
blast
got
and
exposed
BLB.
to the AI from an US AI specialist.
These are some of the pictures which
Our
validate
company
our,
is trying to
our
make
products.
an AI
In the
agent for
middle,
our
you can
R&D,
see
and
Arise
we are
Dhani on the
working
right
on
side
building the
gave
data. Historic
8.3
data is
tons
very
per
important
hectare.
for us. So
And on the
I
left
feel
side, the
whatever
other
I have
cultivar
learned till
gave
now
only
and whatever I
1.7
have
tons per
experienced
hectare
with a limited
under the
exposure to that,
infestation. This
AI
is a
will
farmer
have a
field
lot
photograph,
of
real time.
solutions to us.
Similarly,
with the
you
quickest,
can see other photo where
solutions
Arise Dhani
from
DT,
our
Arise
historic data and
64
future
gold
model
is
building.
crop is standing very well.
AI can help us
But
in
other cultivars cultivar which are not having
in
resistant
a much
gene
better
are
way in the coming
affected by
days.
the disease
Because
and
most
pest.
of the agricultural science is built on vast
Now, what is the economic
amount
contributions of
data.
disease-resistant
For example,
rice
if you
hybrids
take
to
your
the
initial
regional system?
varietal trial, the amount
Here,
of data
we can
that
see
in
over the
that
past
varietal
10
trial
years,
is so humongous
our rice
that
products have
it will be
given
humanly
a
impossible
better
to assess
in
the
return to
variable
the farmers
variation and
compared to the
means
other cultivars
to
when
judge
grown
the
in a
right
disease-prone
variety as a
geographies.
superior. So I feel that AI will be a very
And this is the economic
great
value of
tool
the
in the
disease
coming
resistance in rice,
for
which impact across the yield
developing
levels,
new models
where we
and
see
identifying or
in
decision
3 to
making in
16%
breeding.
yield loss.
With this,
Still, if
I
you
thank
grow
US
resistant
Harvard
products,
and other organizations
farmers
for
can
giving
get
me opportunity.
better returns.
Sir, thank you for me. I feel very proud
These
to
are
be
the
sharing with you
conclusions now.
platform.
It's
The
my
first is
pleasure also,
integrated
sir.
molecular
Now
tools make multi-stress resistance practical.
I now request
Digital phenomics
Dr.
accelerate
Papagoda Patil, who
accurate
is a senior
selection,
rice breeder in Advanta Seeds.
and collaboration is the
give his
key
own
to
perspective.
scale.
Okay, sorry. Okay, no,
Thank you
go
for
ahead,
your
sir.
attention. My contact details are here. If any questions, you can send it
Okay, sorry.
across.
Chandrasekhar from Kotiva.
It's okay, sir. No. So, I think.
Yeah.
Thank
We
you.
are
Thank
like
you, Dr.
fortunate,
Chandel, for
again, as I
excellent
said, like, I am
presentation.
also alumni of this
And
college.
you
Coming
have
back here
covered
and talking in
various
front of
aspects
especially
on
students
rice,
is very
what we
encouraging.
should do in India
so,
and globally.
From
and
morning
what are
we
the
saw like
options
multiple
available with
presentations
us,
about
and
the
giving
disease
little
emerging
bit,
situation and the
information
new tools,
about
how we can
your company's
use
work.
those to
That's
effectively
to
monitor, breed for
in a
the
very
resistance
confidential manner.
and the new
I was
question is about
discussing with Dr.
durability.
Saab
I'll just try to
that,
address each one of those things.
What are
Like
the
one,
genes
like,
which you have deployed
most of the time
in
we
various
discussed about
cultivars?
deploying
Probably
these
you
major
don't
effect
disclose.
QTLs. I think
So
which
anyway, it's
is
your
progressed
policy,
very
but
rapidly
I think
and
students
especially
or
on
anybody
rice.
can ask question.
I always think like
Dr.
in the
Saab,
insect world,
Dr.
Drosophila,
Krishna
and in
Kumar.
the plants world, we always refer to Arabidopsis. But on a cultivated species, rice is having so much information
presentations on rice.
generated in like
We have
multiple
a
million
tremendous
base
post-harvest loss.
GBs, you can
After
say. That
rice
much
is
data is
harvested.
available on genomics,
And most of the
phenomics,
time,
everything is there. How to
our
put that
products
data
or
into
our,
effective
rice
use and make
exports,
meaningful decisions
whether it is in broken
for
rice
breeding
or it is in
is
the
actually a
other
challenge if you see
formats.
if you, that much data is
Often
available.
get,
That is where these
I
new
think
tools,
some
AIs and
effort
all
now
those things will
slowly
be
should
helpful
start
to make that
with
right
respect
candidates
to
because as a plant breeding
breeding
students,
those
we know that
genotypes,
right,
which can
the
also
simple genetic gain what we
confer,
make is time and cost.
post-harvest
Making those decisions
resistance
in time
whatever
and
is
putting
the-
it
because
in the
we
cost
almost
effective
take
manner
it to
is
the grain, and it's a very sad story.
sometimes
to see
we
that
always think that
if
it is
our
only
product
private,
is
but
rejected
it's
both
happening
within
everywhere
and
because
outside.
resources are
I
infinite.
don't think,
So
in my view,
we
that
need to
kind
make
of
those
an
decisions
approach has
in right
been
way.
very
So
intensely done.
market assisted
But
back
since you
as I
said
said, like
it's a
multiples we have seen.
institutional
One of the area which
collaboration,
I mean, like
I see that
we have
where
significantly
if they
developed
can
and we
join
are seeing a
hands with
benefit of
others
that is
and also
especially
see
in a
if we can take
self-pollinated
it
crop
forward,
rice,
it will be very,
genomic
great
selection.
advantage to our country
Because we are
since
trying
we
to
are also
make
one of the
a
largest
self-pollinated
producers
crop
and
into
exporters.
hybrid crop.
Thank you,
And
sir.
there
Thank
are
you very
challenges
much for
like,
your
for example, I
comments.
want to
Thank you,
evaluate
sir.
10,000
This-
test crosses.
This
It's
is very well
difficult to
commented,
generate
and
those many
this is
test
need
crosses,
of the
put
hour.
those in field and
But
evaluate.
I would
So
also
that
like
is
to say
where
that if we
your,
harvest the
genomic
grain
selection
at the
tools
right
will help significantly
moisture,
and make lot of
we can
predictions.
still reduce
and
all these
generate
post-harvest losses.
thousands of
But we have
recombinants
to work
and
together
advanced
on these
rice recombinants.
technologies.
Otherwise, we